Hello and welcome to Chapter 21, Part 1, The Evolution of Populations. So this chapter is going to emphasize genetic variation, phenotypic variation, what's called the Hardy-Weinberg equation, which is really just a way of quantifying genetic variation. And then if you can quantify it, then you can look for changes over time, for example. And then what are those forces of change that would maybe change genetic variation over time? Now, just a reminder here, this is something we've talked about previously, this idea that populations evolve. Organisms don't evolve in their lifetime. They might, you might change over time, but what evolves from that perspective of, biological evolution, that's the population. And you've seen this simple example before where we have organism X, whatever it is, where here yellow is selected against, blue is selected for. We're saying the yellow ones are less tolerant to UV radiation, and over time with that exposure, the population will change so that multiple generations later, you see more blue-colored individuals. All of this change is contingent on variation. Let's say this whole population was just these yellow-colored organisms, and they're selected against, and if strongly enough, that might mean they're wiped out. You need variation to be there for natural selection to work on. And so when we look at variation phenotypically, like these ladybugs here in this picture, that expression of the phenotype is dependent to a large extent, or at least some extent, varies depending on the trait we're talking about, on genetic variation. And of course, what is genetic variation? Those are those different kinds of alleles that we might see for different genes that maybe are different between different organisms. One of the things that is very useful to look at is to look at overall heterozygosity. Now remember, if we're looking at a particular gene, an individual is heterozygous if they have two different alleles. If they have the same allele, like we see here in this picture, that means they are homozygous. And the ones that have different pairs of alleles, those are heterozygous. Now, why would looking at heterozygosity be useful for understanding overall genetic diversity? We can illustrate that with this example here. So if we have a gene that only has one allele, that means 100% of the alleles are allele P, we're just going to call it that. The expected heterozygosity would be 0, right? Because you can't get anything other than that P allele. Every individual is going to be homozygous. Let's say there are two alleles, and we're just going to assume they occur in equal amounts in the population. So we have P and Q here. If that's the case, then heterozygosity is 50%. And this might remind you back to our Punnett squares in genetics, that 50% of our individuals out of a cross would be heterozygous. Now, what if there are three alleles? Again, now remember, we're thinking at a population level here. There's three alleles in the population. Each individual only gets two of them at any given time, obviously. And we've got P, Q, and R. The expected heterozygosity here is 66%. Let's just keep going here. We have four alleles. That, if they're all the same frequency, 25% each, 75% heterozygosity. So you see heterozygosity increasing when you have more variation. You have more, an increasing number of different alleles for a particular gene. So that's why this is useful. So when we're looking at genetic diversity, nowadays we can generate DNA sequences very readily. And we can look in what we're looking for are differences in nucleic acid sequence. And so there's a substitution in one allele versus not in another. Then we'd say, okay, we've got two different alleles here, or whatever it is, 3, 5, 6, 7, 8. Now, those differences, a lot of times they occur in non-coding regions. So here's this gene, the dark blue areas are exons. Remember, those are the expressed regions, and then the introns, which are spliced out. Notice you have these orange lines. Those indicate substitutions, and a lot of them occur in introns. There's a lot of intron sequence in here. And so just by chance, you might see more of these in introns. The other thing too, right, is if you have a substitution in a gene itself, that might have an impact on what the gene is. So here we have that silent mutation. Lysine is still lysine. But you could have, a substitution that creates a stop codon. That's really going to change what that gene product is if you have a stop codon where it wasn't before. Now, a lot of times mutations like these silent mutations might occur, or even this missense mutation that's called a conservative missense mutation. We've changed the amino acid, but these two amino acids are fairly similar. So it's conservative in the sense that this protein isn't probably going to be altered that much. Whereas if you had a non-conservative substitution here, this is a very different amino acid from lysine. Now that might impact the say, structure or activity of the protein. But it depends where these things occur. If this is a substitution that occurs in what will be the active site of an enzyme, that's probably really critical. If it's a substitution that occurs that results in a different amino acid just somewhere out in the structure of that protein, maybe it doesn't matter. So some of these can be useful for understanding whether we're dealing with multiple alleles, and even looking at these non-coding regions like introns is useful. Of course, it's not just genetic variation that gives us different phenotypes. So the environment might play a role in what phenotypes are expressed. Now, this is really important that, you may have phenotypic differences, which you might think, well, natural selection's acting on that phenotypic difference. You say these different caterpillars here, I don't know, let's say the birds can see these ones on the left more readily than these ones on the right that look a little more camouflaged. You might think, well, that's going to change the frequency of whether you see one phenotype or the other. The issue with this example is these phenotypic differences are based on diet, not genetics. So if the birds are eating these caterpillars over in panel A here, it doesn't matter. It won't change the genotypic frequencies over time. Remember, for natural selection to work, you have to be dealing with something that's heritable. And in this particular example, not heritable. But it does highlight the importance of environment. So you've got genetics underlying, but the environment can also influence, obviously, the characteristics that are displayed, if you will, in these different organisms. So where does genetic variation come from, right? So, if we've got two alleles, think back to Mendel for, say, flower color. How do you get a new allele, a third allele? There's a variety of ways that we might see this occur. Maybe the obvious one would be a mutation, this is something we've talked about at length. You get a substitution that maybe changes the protein, now produces a blue flower, for example. We saw where a lot of variation is introduced in sexual reproduction. So think of meiosis and fertilization. And then there can be gene duplication events that create new copies of genes that then can diverge from the existing copies. And so let's start with mutation. This should be something you're already fairly familiar with. As soon as you have a mutation, say a substitution, and even if it's neutral, that means it doesn't affect the outcome of that protein, for example. That is a new allele, right? It's genetically different. Again, whether it actually produces something that can be selected for or against, that's another question. And so mutations occur just at random or from environmental interactions, chemicals, UV radiation. And so that's how mutations might occur. A lot of the time, a mutation probably doesn't matter if it occurs in introns, like we talked about already. Only really when a mutation is harmful might it be, say, selected against. And then maybe a mutation might actually be beneficial. So in this example here, we see this one mutation occurring, this advantageous mutation in red. And then through selection, in this case, it's called a selective sweep, you're selecting for individuals that have that mutation, and say it's beneficial. You're losing some of the genetic diversity, but you're also dragging along, if you will, these other mutations, which may or may not have been beneficial or have a negative impact, but because this other one is associated with it in the same gene, you get these other mutations coming along as well. All right, should be an easy one. A mutation that's neither harmful or beneficial is termed what? All right, yeah, we've talked about these before. A silent mutation, you've got the genetic change there, but ultimately the protein wasn't changed. So that's a silent mutation. And that would be considered a form of neutral variation. So we have technically a new allele when you have that mutation occur, but ultimately it doesn't change the phenotype because that protein, say, still has a lysine in it, and it had a lysine in it before. So this is going to be neutral from a natural selection perspective. Neutral mutations might also occur, again, back in, say, introns. So they don't really have an impact on the phenotype. They're just happening there. Or other parts of the genome that aren't expressed. Those mutations can occur. They may be useful for understanding, say, relationships or evolution between different organisms. But those are effectively neutral from a natural selection standpoint. So let's layer in meiosis here now, thinking, sexually reproducing species and where that genetic variation comes from in them. So here we have allele 1 here. And in the second generation, you have these two, individuals. Here, a mutation occurs. So now there's a difference between those alleles. Here's actually a second mutation that occurs in generation number 3. So now these individuals come together and mate, producing this individual here, which again, through meiosis, during gamete formation, now having these two different alleles, same gene, but now this individual is heterozygous, having allele 3 and allele 1. And when crossing over occurs during meiosis, you actually have now created yet another allele. Here's a fourth allele because as a result of crossing over. So sexual reproduction is definitely a key driver of genetic variation. And this is something we've already seen and talked about. So that crossing over that occurs in meiosis is critical. Independent assortment a little later in meiosis. And then, of course, random fertilization, the idea that individual gamete could potentially, theoretically, fuse with another individual gamete in billions of random potential combinations. Something else that we see that generates variation, which I've mentioned before, is gene duplication. So this is a nice figure. I really like this. So we have the gene with this ancestral function that's shown here in blue. A gene duplication event occurs. There's many ways that can happen. Let's just say it does. And now you have two copies of this gene. So maybe here, conservation, both copies are conserved. Both are expressed. They retain their ancestral function. Maybe one of the copies becomes non-functional. It's grayed out here. Maybe between these two copies, parts of them acquire or generate a new function, maybe through mutations that occur. Here's something called subfunctionalization. So you have parts of each of these duplicated genes that lose their function, but they still have parts of the ancestral function, and together they can work. And then, of course, this is the really interesting one at the bottom, neo-functionalization. The second copy starts to generate a new function. So we have a new gene, really, that has diverged from the original copy now that you have two, and it has a new function. Now, a cool example of gene duplication, and there's many, but in humans, are odor receptors. So we have many different genes that are responsible for odor reception, and that's really important, distinguishing smells. And you can see highlighted on this human genome here in red, or maybe you can see it, these little red spots. These are places in our genome that produce genes that are important for odor reception. And a couple of examples here, region 1q44, that's down on the end of our chromosome number 1. That actually has 29 genes. So not only are there a bunch of different regions on our chromosomes, but there are multiple genes at each locus, each place. Here on chromosome number 11, this one has 56 genes here, down on, or actually here on the other end of our chromosome. And so these are a result of gene duplication events, and really important in, say, the evolution of odor reception in humans. Okay, so quick review type question. Why don't similar genotypes always produce the same phenotype? All right, yeah, hopefully an easy one. It's because the environment matters too. Genotype is very important, but the genotype interacts with the environment to produce that ultimate expression of those genes, which is the phenotype. All right, so let's now look at quantifying patterns in genetics. So R.A. Fisher here is known as the founder of population genetics. That's what we're really talking about here. And kind of like with Mendel, who combined fields to make a really important leap forward, so did R.A. Fisher. He takes statistics and genetics and what is his understanding of evolution and puts these things together to create this field of population genetics. So when we're studying population genetics, of course, it's really important that we understand what a population is. So a population would be a group of interbreeding organisms of the same species in the same geographic area. We'll talk more about what a species is in the next chapter, but a species, of course, is a group of individuals that can exchange genetic information and share alleles. And so the critical thing here is interbreeding, and we'll see how that's maybe not always applicable to every species, but that is fine for the moment right now. The other critical thing here is understanding what a gene pool is. And so this is all the alleles in all the individuals in a species or population that we're interested in. Now, again, when we talked about genetics, we really focused on, well, individuals have 2 alleles, and that still holds. But if you start to look at the whole population, maybe you say, well, there's like 16 different alleles in that population. Well, individuals only get 2 each, but it's important to understand how much genetic diversity is there. An example of population, or 2 different populations here, are with caribou. Now you'll see the range of these 2 different caribou herds. They do overlap, but we consider we consider them the same species. They have the potential to interbreed, but they do tend to only interbreed within their own population. So even though there's overlap, we have 2 distinct populations. All right, so another thing to keep in mind, thinking back to genetics, we've got variability in a population, genetic variability, and parents can pass alleles to their offspring. They can obviously only pass the alleles that they possess to their offspring, and you still might get different combinations of alleles than, say, that were in the parental generation or in those particular parents. And then you say, well, what happens to the alleles that, say, aren't in this family? Again, you got to zoom back out and think about it at a population level. Here is where we see those other alleles if we look at enough individuals. And some of those alleles get passed in a certain family, some of them don't. But at the population level, they make it to that next generation most of the time, and if you have a big enough population. When we see the consistent passing of alleles so that we would see the same proportions of those alleles generation to generation, we would consider that a genetically stable population. And we'll see how that's useful as a baseline for understanding when a population isn't stable and what then might be happening. So if we want to understand the allelic diversity or genetic diversity, we need to do some basic math. And it's really helpful to calculate what's called an allele frequency, which is just how often is that allele there. And you can look at it at a population level and just count up if you could. That might be based on phenotype or genetic sequencing. I'll talk about several examples. How many copies of that allele are present over how many alleles are there? And so let's look at an example. So here's where we're going to do some DNA sequencing. So we'll get we'll know exactly what alleles we're dealing with. We're looking at this particular gene, alcohol dehydrogenase, in 50 fruit flies. So this is important. Now, each fruit fly is going to have 2 alleles, right? So our denominator in our equation will be 100 because each individual has 2. There's 50 individuals. 2 times 50 is 100. And so what do we find? We find that 70 of the alleles have an adenine residue at a particular site. Think back to substitution. and 30 have a guanine, a G, at that same site. So that allows us to say, well, okay, we've got an allele with an A and an allele with a G, and we can then divide them up. And again, we're just counting here. So the A allele, there's 70 of those out of 100. This is 0.7, that's the frequency. And we often will convert these after to percentage. It's maybe sometimes easier to talk about that. Now, remember, if you're converting to percentage, this is what this really means is 70 over 100. And so if you were to use this in a later manipulation, you probably want to go back to this frequency, which is 0.7, rather than taking 70, for example. This is not really 70. This is 70 over 100. Our other allele is, 0.3 or 30%. Notice too, if you've got two alleles, they have to add up to 100% or 1. That's really critical. So once you figure out 1, you can easily figure out the other one just by subtracting from 1 or from 100% here. All right, so it's your turn now to do this. We've got a survey of salmon in the Columbia River. We collected 100 fish. And we're looking at a particular gene. Remember, each individual has two alleles. So if we've got 100 fish, what's that denominator going to be? And we're going based on the data here. We have 14 salmon that are homozygous big R, 52 big R, little r, 34 that are little r. This is just a counting game. So count up how many of these alleles you have and use the correct denominator to determine what's the frequency of big R. All right, so it's important to be able to work through these, and hopefully you got to 40%. And how do you solve this if you got stuck and you were just guessing? It's not that complicated. Think of which of these, where do we see a big R allele? In these 14 salmon up here, and each of them has 2, so there are 28 big R's in these homozygous fish, and there's only 52 R's in these heterozygotes. In these homozygous little r, there's no big R allele. We don't need to account for those at all. So we just add these up. 28 plus 52 is 80. And remember, there's 200 alleles in 100 salmon. So it's just 80 over 200, which is 0.4 or 40%. All right, let's continue with another example. In this case, we have an obvious phenotype that we can link back to a genotype. So we're using the R allele here again. In this case, we've got flowers that are red or dark pink here, and those are homozygous big R. There's lighter pink is big R, the heterozygotes, and little r are white. Now, you can just go out into the field and count these up. So this is kind of convenient when you have that phenotype-genotype direct link. There's no dominance recessive situation here. We have incomplete dominance, so we can just count them all up. And so here we do a survey, we count 1,000 plants, and here's what we found. Now, from this, you should be able to figure out what the frequency is of these different alleles. Again, it's just counting the big R's. So let's take our homozygous plants. There's 90 of those. That means there's 180 big R alleles, right? Each one has 2, so it's just 2 times 90. The heterozygotes, there's 420 of those. There's only 1 big R allele in each of those, so that's 420. And so then we can sum these up. So we have 600 big R alleles. We have 1,400 little r alleles just based on our summations here. Remember that works out. That makes sense because we have 1,000 individuals and that means 2,000 alleles. So now we can figure out the frequency. Here, 600 divided by 2,000 is just 0.3, or 1,400 divided by 2,000 is 0.7. Again, let me remind you, notice that the frequency of R, big R, and the frequency of little r, that equals 1. They have to add up to 1 or 100%. Because those are the only 2 alleles in town right here. And so together they must add up to 100%. All right, so this next idea, let's see what happens. Pollinators are flying around. How many plants will we have next year that have red or pink or white flowers? What's going to happen? Or how could we predict that? Well, if we know the frequencies of these alleles, which we do, we can just plug those into a Punnett square, basically. So let's do that. Look here. So if we say, well, individuals just by chance are going to pass, there's 30% chance they'll pass on a big R allele, 70%, 0.7 frequency of little r. And you can do the same over here on the other side of our Punnett square. And then we're just going to put them together. Think fertilization. So the odds of getting a big R in the next generation, it's just 0.3 times 0.3 or 0.3 squared. Little r, it's 0.7 squared. Now, whether we have a heterozygote or not, there's actually two ways to get that. So it's just 0.7 times 0.3 and 0.7 times 0.3 again. But you can do that in two ways. So you could really just say 2 times 3.3 times 0.7 if you wanted. It's a shortcut. And this can be expressed mathematically if we denote the frequency of big R as P. These are common variables that we use, P, and little q. And notice again, so big R, that's just P squared. Little r, that's just Q squared. And again, the heterozygotes, there's two ways to get those. And so we could just say 2PQ. Now, remember, counting up all the individuals, that has to equal 100% or a frequency of 1. And so if we put these together now, P squared plus 2PQ plus Q squared, that has to equal 1. Just go back to our plants that we were counting up. It can't equal 80%, right? No, it has to equal the total of 100% because that's how many plants we're counting, whatever that happens to be. So knowing that means if you can solve a couple of these, you can then figure out the other one just by subtraction. So this idea that we can represent mathematically, thinking about that Punnett square, just bringing alleles together, that P squared plus 2PQ plus Q squared, this is called the Hardy-Weinberg equation, and it's based on the Hardy-Weinberg principle. The idea that genes occur as pairs of alleles and that we can simply count them up and determine how common they are. Now, in our example with the flower color and the previous examples with the fruit fly genetics, we were able to identify exactly how many alleles there were just by counting them. Sometimes we don't know, and that would be an example, say here, of the coat color in these cats. We know how many individuals, say, are little b for these white-colored cats, because we can just count them up. But these black-colored cats, which is 84% of our sample here, we don't know, are they homozygous, big B, or heterozygous, big B, little b? We don't know, but we can determine the frequency of the little b allele based just on the white cats, and then from that determine what the frequency of big B is. So if 0.4 is little b, 0.6 must be big B, right? Because if 40% of the alleles are little b, then 60% must be big B. And then once we know that, we can plug these into our Punnett square again, just recognize that the likelihood of getting a big B is just P squared, 36%. And then our heterozygotes here would be 48% together, right? 24% each. And that's just using this equation. So we can use this to predict what's going to happen in future generations given a known frequency of particular alleles. Or we could say, well, here's the frequency of these alleles, and our expectation would be that, say, 16% are these white cats, 36% are homozygous black cats. And if our sample then actually differs from that number significantly, that something might be happening in that population that's pushing the frequency of those phenotypes really away from our expectations based on allele frequencies. All right, so let's work through an example together here. We're going to use Mendel's pea plants with these purple and white flowers as an example. We're going to use the allele F. Big F is dominant over little f. That's if you've got one big F, you have a purple flower. So that's that just dominant recessive trait. And we go out and count 100 plants, and 64 of them have purple flowers, 36 have white flowers. Now remember, we don't know in the 64 here, how many of these are heterozygous and how many are homozygous? What we do know is how many are homozygous recessive, the 36 here. And so from this is where we can start. All right, so we know that there 36 of the 100 plants that we counted, those have the genotype FF, right? Little f's. That can be represented in our equation by q squared. That was that bottom corner of our Punnett square. And so we don't know what the frequency of little f is, but we know that q squared is equivalent to this number here. And so q squared equals 36 over 100. And so if we want to know what q is, which is equivalent to the frequency of f, we would just take the square root of both sides of this equation. And this is set up easily, so you can take the square root of 36, it's just 6. And then ultimately we have 6 tenths, which is 0.6 or 60%. And you really want to focus on these frequency numbers. If you start throwing these percentages in or mixing these in, you'll start to get some wonky data here. All right, so what's the frequency of the other allele, big F? So the frequency of big F, that equals P. Now, we only have two alleles here. If Q is 0.6, then P must be whatever is left, right? And if, P plus Q equals 1, if Q is 0.6, then P must be 0.4. So we can just say 0.6. 1 minus 0.6 is 0.4. All right, so now we have both bits of data that we need. How many are heterozygous? How many plants are heterozygous? Remember, we didn't know they're just purple flowers or not, and we don't know whether the purple flower plants are heterozygous. How many are heterozygous? How many are homozygous? It's 2pq, and we know these numbers now. 2 times p is 0.4. And Q is 0.6. And so this is equivalent to 48 over 100, or 0.48. All right, so we know a couple of things. We know this frequency of the homozygous recessive is 0.36. We know the heterozygotes over here, homozygotes up here, heterozygotes over here is 0.48. How many are homozygous dominant? That's just p squared, and so that is 0.4 squared, which equals 16 over 100, or 0.16, or 16%. Now these should all add up too, right? So if 0.36 plus 0.48 plus 0.16 equals 1, then you're on the right track. The other thing we could have done here is, knowing 0.36 and 0.48, we could have just subtracted those from 1 and we'd end up with 0.16. All right, and so here we see those numbers worked out in text. You can come back to this and look at it. Really, it's fairly simple. If you know that base equation and you know that the components of it, whether you're just looking at the alleles themselves, P plus Q, that has to equal 1. And then at the genotypic frequencies, P squared plus 2PQ plus Q squared, that also has to equal 1. Together, each of those has to add up to 100%. Then you can figure out really any of these problems. And so here's an opportunity to try that out. So we have a mouse population, 200 mice. Brown fur is dominant, so we have a dominant recessive situation. You have 120 mice with brown fur. That's the dominant trait. What's the frequency of mice that are heterozygous? Now remember, start with the gray fur. Those are the ones we know the genotypes for, and work your way toward how many are heterozygous. All right, so again, we know how many are homozygous recessive. That's 80 out of 200. That equals that 2 squared number. You could change that to 40 over 100 if you want for convenience. So if you want to figure out what's the frequency of little f, that's just Q. So that's the square root of Q squared. Once you have that, now you can figure out, well, what's P, because just 1 minus Q equals P. Now you have both the frequency of P and Q, and so the heterozygotes is just 2PQ. Plug in the numbers, you get 0.465 or 46.5%. All right, so let's continue this same example so you can use some of the numbers you already have calculated. What's the frequency of mice that are homozygous dominant? All right, so we already have some numbers to work with here. It's just P squared. We know what P is. So 0.368, or take that and square it, you get 0.135, which is 13.5%. Another way to approach this would be, we already know the frequency of the homozygous recessive and the heterozygotes. You could just subtract that from 1, or if you're working with percentages, from 100%. And you would end up with, in the same place here, 0.135 or 13%. So understanding how to do some of that math in terms of allele frequencies and figuring out what our expectation would be given those allele frequencies is really helpful. And Hardy and Weinberg had, came up with this principle, and hence it's named after them. And interestingly, they came up with it independently. And then sort of discovered that they were each working on it, and they collaborated. They decided to work together. So this is not one of those situations where one person sort of grabbed for the credit and the other person was lost to history, but they collaborated, and both their names are on this. So taking this genotypic type information in allele frequencies and genotype frequencies, they ask, well, what would happen for this to stay stable over time? And they said, well, there are five things would have to happen for these genotype frequencies to stay the same over time. And the assumptions they made were these 5 things. There would have to be no mutations, right? Because if you have a mutation, you have a new allele. That definitely changes your allele frequencies. You had 2 alleles, now we have 3. That's going to change your numbers. No migration. We'll see why that's important. Mating is random. So in a population, if certain individuals are mating with other certain individuals, that might change genotypic frequencies. The population has to be large, because if you have a small population, it's more susceptible to random chance events that might change genotypic frequencies. And there can't be selection for a particular genotype, for things to change, right? If back to our colored dot example, if there's selection for, say, the blue colored organisms, that's going to change those allele frequencies. Those are going to become more common over time. And so it's good to start with that null hypothesis or baseline, but we can really say, well, how likely is this to happen? Really, these things are happening. Mutations happen, selection happens, mating isn't completely 100% random. So that means change is happening. And this is the foundation of evolution. This is called microevolution. We're looking at the genotypic population changes. So let's apply that to a human example here. And we're looking at a condition called PKU. And we say, okay, this is in Hardy-Weinberg equilibrium in the United States. We're looking at the US population. And if somebody has PKU, you will know it. And so you can count the individuals that have PKU. Now, the other thing we might be interested then is, well, how many carriers? What proportion of the US population is a carrier for PKU, this recessive condition? So they have the allele, but they don't have the condition, but they potentially could pass that along to their offspring. So if we assume mutation rate is low, and that's reasonable here, mate selection is random. Now, with respect to the PKU allele, then that's probably true. It's not some sort of outward phenotypic characteristic that might influence mate selection. Natural selection only acts on rare homozygous individuals that don't follow dietary restrictions. So PKU is a condition where you can only consume certain types of food. And if you follow those, you're going to be fine. You're going to reach an age where you can produce offspring. The population is large. That's reasonable. U.S. population is, hundreds of millions in size. And then this comes into the idea of migration, that if you have people moving in or out, that the frequency of those alleles in other populations is similar. So we're gonna use those assumptions as we dive into this. So the occurrence of PKU, the condition, is 1 in 10,000 births. So we can take our 1 in 10,000, that's our Q squared number. And that allows us to calculate the frequency of the PKU allele, which is 0.01. If we have that, again, you've done this already. You can figure out, well, what's the frequency of the non-PKU allele? It's just 1 minus Q. So in this case, P is 0.99. Now we can calculate everything else. We have P and Q. We can plug it into our equation. And so the odds that someone is a carrier is 0.0198, which is about 2% of the US population. So, pretty rare. So here's how we can apply this to a real world type of question. And this might be important if there was maybe even a family history. You could track the trait even closer than that. And figuring out what are the odds that someone might have a child, a random couple might have a child that has PKU. So this is a very useful tool. Okay, so what happens when these assumptions are not met? We're going to see deviations in those expected genotypic frequencies. And so when we crunch our numbers and go out and count individuals, and if we see differences from what we'd expect, that suggests that change is happening. And this is much more likely than a population being static over hundreds of generations. And what is going to cause those changes, we'll just flip those assumptions on their head. Remember, one of assumptions, no mutation. Well, if mutations occur, that is going to drive change. No migration. If there is migration, we call that gene flow. So migration with mating, really. That mating had to be random. Well, here, change will happen if there's non-random mating. Certain individuals only mate with other certain individuals. Genetic drift. This has to do with random events that can happen in a population. And if a population is small, random events have a bigger impact than if a population is huge. And then, of course, natural selection in general, that will drive, say in this example, the birds eating the yellow caterpillars. That's going to push the genotypic frequencies toward these green pigment producing caterpillars. And so these are our agents of evolutionary change. We'll talk more about those in the next part.